Good evening, everyone. We're going to get started shortly in a few minutes. I'm just having everyone join us. Just give them one more minute. All right, so we're going to get started. Thank you very much for joining for today's Grand Rounds. I'm actually hosting the Grand Rounds tonight from Washington, and I'm here for attending the TCT. Today is a very important day. There were 3 late-breaking clinical trials in clinical cardiology. One was on the TAVR, and now asymptomatic patients with aortic stenosis. TAVR was found to be effective to watchful expectancy. And the remaining 2 studies were in the similar leagues. But what is very critical to recognize is whenever the heart is under stress, what makes it what keeps it resilient. So we're going to talk today and switch gear and talk about myocardial metabolism. And there is very exciting pathways that we're going to learn. And for today's specific CME, I'm going to introduce our CME slide sets so that you're able to take your MOC and CME credits. So you'll have your lecture again posted on the website, on the YouTube, where you can view it. And you can earn your CME credit. You can go now and text 25659 to the number 888-816-489 for your Cloud CME platform as a mobile text, and be able to get your CME credit for the session. And for physicians, for the MOC credit, you have the link that will be displayed back again. This link is here. It will be displayed back again. And the room code is Future 2024. You have the barcode displayed here. You can scan it and you can complete those steps even now for your MOC points. With that note, I'm going to bring in Dr. Yanmala to introduce our speaker because the speaker is from her section of clinical research and AI innovation. Thank you. Navina?
Thank you, Dr. Sengupta. It is my honor and pleasure to introduce Dr. Ahmed Clanton, who joined Rutgers University in the month of September of this year. He's a tenure-track assistant professor in the Department of Biochemistry and Molecular Biology, with joint appointments in Division of Endocrinology, Nutrition, and Metabolism, as well as our division in the section of Clinical Research and AI Innovation. Welcome, Dr. Clanton. And Dr. Clanton brings a wealth of expertise in biochemistry and molecular biology, focusing on how metabolic pathways influence cardiac health and disease. His academic journey began with a bachelor's in clinical laboratory sciences from Kingswood University, followed by a master's from Georgetown University and a PhD from Cornell University, where he conducted groundbreaking research on glucose and glutamine metabolism in cancer. Following this, he continued his research as a postdoctoral fellow at the University of Utah until he joined Rutgers, where he investigated lactate metabolism in cardiac health under the mentorship and guidance of Dr. Jared Ratter and Dr. Stavros Drakos, who is a heart failure cardiologist. Dr. Clanton's research achievements have earned him several notable accolades, including NIH NHLBI K99/R00 Pathway to Independence Award, and also the Evelyn Brunger Award for Postdoctoral Excellence. His research mainly explores the metabolic pathways influencing heart failure, focusing on lactate and pyruvate metabolism. His contributions were published in high-impact journals like Cell Metabolism and Journal of Clinical Investigation Insights as well. These publications have shed light on metabolic flexibility as well as resilience in cardiac tissue. Dr. Clanton has also presented at many national and international conferences and several keynote symposia, including topics on heart failure and other heart diseases. Please join me in welcoming Dr. Ahmed Clanton to share insights from his latest work, which examines the role of mitochondrial pyruvate metabolism in heart health. Thank you, Dr. Clanton, for joining us.
Thank you. Thank you for the invitation. Can you hear me well? All right, great. Thank you. So thank you so much for the invitation. It's great to give this talk. I'm very excited to share some of the work we've been doing to clinicians. And I know one of the great common interests among clinicians has always been metabolism. Just kidding. I know this is a little bit of a boogeyman for many clinicians. And believe it or not, when you look at a map of metabolism, such as the one we see right now on your screen, this was made to simplify metabolism for medical students and has since turned into a very complicated, simplified representation of metabolism. So more recently, people have tried to further simplify this by making it look more like the subway train map. So every pathway is just a different color, and it looks it's a lot more pleasing to the eye, very easy to follow, and it makes it more intuitive. Now, the problem with this representation is that it gives the false idea that metabolism is very rigid and one-dimensional. So once you're on a track, you can't really get off the track, and you can only go in one way or the other. Now, in my opinion, a much, much better representation of metabolism would be shipping routes. So this is a really cool website, shipmap.org. If you ever have time, you can actually see all the ships moving in real time online. And what you note here is that the routes are very dynamic. They're not stuck to a specific track, and they can move as needed. And more importantly, if you pay even more attention, what you'll notice is that there are, in very specific locations, there are many bottlenecks where multiple shipping routes converge. And these are often the canals or channels or straits. What we know for most superpowers, for example, historically have known this, is that anybody the Army or the Navy that controls these canals and channels is also the Navy that controls the world. And in a very similar manner, these channels are much like transporters that can connect multiple compartments within a cell to allow metabolites to go from one place to another. And if we can control those transporters, we can really control the destiny of a cell. So perhaps one of the most famous transporters, and one that was discovered by my postdoctoral PI's lab, Jared Rudder, in 2012, is the mitochondrial pyruvate carrier, or MPC. Now, this is an obligate heterodimer made of MPC1 and 2 that localizes to the inner mitochondrial membrane and facilitates the transportation of pyruvate from the cytoplasm into the mitochondria, connecting two very important metabolic pathways in glycolysis and the TCA cycle. Coincidentally, when I joined the lab, one cool observation was that it turns out that the organ with the highest expression of MPC was the heart. But yet at the time when I joined the lab, nobody studied the heart. And I thought this was probably an ideal organ to look into. And it's not, in my opinion, at least at the time, was not that surprising that the heart had the highest expression of this MPC because the heart is always pumping. In fact, as many of you might know, the heart is the first organ that's developed embryonic day 22. It starts pumping by embryonic day 22. It's the first organ that's developed, and it continues to pump, never stops pumping till the day you die. So you require the heart to stay alive. So it's not surprising then that the #1 cause of death on planet Earth is heart disease. And this is true in the US and globally. And I think we probably need to do a better job to communicate this to the public, because I know, at least for me and many of my colleagues, when you ask them what's the #1 killer, they always think it's cancer, but the heart, you're more likely to die from heart disease than from any others. But we don't do a good job of letting people know about that. And one important aspect of the heart to continuously beat, since it's beating throughout your entire life, is that it needs to be very flexible. So it's been known to be a metabolic omnivore, which means it can make ATP or can make energy from very different substrates. So mainly almost 80% of the ATP that the heart generates is coming from fatty acid oxidation, but it can also generate ATP from glucose, ketone bodies, lactate, amino acids, and others. But however, when there's a problem in the heart, for example, during heart failure, when the heart can't pump enough blood or isn't able to pump enough blood to support the body, there is a known rewiring of metabolism where you see increased glycolysis coupled to increased lactate production, a decrease in fatty acid oxidation, and an increase in ketone body amino acids. Now, when I joined the lab, the thought within the field was that these changes in metabolism were mere consequence of wait a minute, was a mere consequence of the disease. And nobody thought that these changes in metabolism could be a cause of hypertrophy or cause of the heart failure. So when and I'll talk briefly about my first paper here that came out in Cell Metabolism, where we found that the MPC was very important for the heart. In fact, we developed these heart-specific inducible knockouts of MPC within these mice. And what we found is that knocking out the MPC was sufficient to induce hypertrophy in these mice. And by week 16, they all got heart failure and died. So they all got hypertrophy and they all got heart failure just by knocking out this one gene, this one transporter. So it was clearly causing the disease. Moreover, if we overexpressed MPC, we could then attenuate this hypertrophy. So here, what I'm showing you is an experiment where we use H9C2 cells, cardiomyoblasts, rat cell, cardiomyoblast-like cells, I guess, where we give them these different hypertrophic inducing drugs, angiotensin 2, isoproterenol, phenylephrine, and in every case, you can see that there's an increase in cell size, showing that there's hypertrophy that is happening. And if you inhibit MPC by this inhibitor UK509, just inhibiting MPC, that also is sufficient to induce hypertrophy. However, if you overexpress MPC on top of this treatment, you remove this hypertrophic phenotype that goes away completely, and these cells never get into hypertrophy. Moreover, we thought, well, this is all cool and awesome, but can we now utilize our knowledge of metabolism to come up with an innovative way to treat the whole disease? So what we know, what happens during hypertrophy and heart failure is that there's a rewiring, as I mentioned, of glycolysis, where you have increased glycolysis coupled to increased lactate production. And because of this, you have an increased expression of this transporter, MCT4. And this is the major lactate exporter. So it exports lactate out of the cell. And normally, it's not very highly expressed in the heart, but during hypertrophy, its expression goes up. So what we did was we developed a specific inhibitor, small molecule, to inhibit MCT4, inhibit this transporter, so that lactate cannot be exported. So while it then accumulates within the cardiomyocyte, it then shifts the balance of the lactate to pyruvate ratio and pushes LDH. And this is the enzyme that is bidirectional, that can make both pyruvate and lactate, and then pushes in the opposite direction to make more pyruvate and then get into the MPC. And maybe then we can rescue the phenotype. And so this is very similar to what happened, if you remember, a few years ago during COVID. There was this, the Ever Given ship that shut down the Suez Canal, and the world economy didn't just collapse. All what happened is that ships just found a different way to get to Europe or vice versa. So there's always going to be a different route to get there. So because, again, metabolism is very dynamic. So to test this, what we did is that we implanted mice with these osmotic pumps that pump isoproterenol to induce hypertrophy or vehicle, and then gave them orally gavaged them with this VB124 inhibitor to inhibit MCT4 or control. And what we saw there was that when you give them isoproterenol, the hearts get bigger. However, if you on top of that also give them the VB124 and inhibit MCT4, inhibit lactate export, inhibit the MCT4, this hypertrophy was completely mitigated. So that even though they were still being pumped by isoproterenol, that was not sufficient to induce hypertrophy as long as you didn't let lactate get exported. And we took this also to another model of heart injury, which is ischemia or perfusion, which is the second paper that came out recently. And we also, in that model, showed that that inhibiting lactate export also increased salvageable tissue and decreased necrotic tissue within those mice and increased survivability of these mice. So in two different models, we inhibited lactate export, and somehow this turned out to be very beneficial to the mice, to their heart specifically. And I encourage you to go read. I don't have time to go to the details of both of these papers, but nonetheless, this intrigued me very much because it was not really expected that inhibiting lactate would be so beneficial. And now lactate is a very important metabolite, but it's greatly misunderstood. In fact, if I asked just random people on the street what they felt about different metabolites and which ones were good and which ones were bad, you would probably see something like this, where lactate would always come at the very bottom because everybody thinks of it as being very toxic, that it causes problems with your muscles and that it's not good for you. But in fact, that is not true. Now, the reason people think that is because historically, Otto Meyerhoff, Nobel Prize laureate, did this this experiments back in the 1920s, where he put frog legs in this airtight jar, and then hit them with an electric current to get them to contract. So these legs kept contracting, and at some point they stopped contracting. Now, when they stopped contracting, he opened this bottle, looked at the muscles, and saw that they were bathed in lactate. So then he made the assumption that this lactate was causing the problem, that the lactate was causing was inhibiting the ability of these muscles to contract. But it turns out later on that when people repeated this experiment, that the problem wasn't lactate, but it was the lack of oxygen. So if you do this with circulation, with oxygen, you never get this problem. And lactate does not cause any problems to your muscles. In fact, lactate is a major fuel for multiple organs. Lactate is the second most abundant metabolite in your circulation, second only to glucose. Glucose is at 5 millimolar. Lactate is about 2 millimolar. Everything else is more or less in the micromolar. And this beautiful work from the Rabinowitz lab showed that the heart consumes lactate and can feed the TCA cycle and many other organs as well. But more recently, they showed that the heart is actually the highest lactate consuming organ in the body. So among all organs, lactate consumes the most, the heart consumes the most lactate within your body. So all of this is really cool, but how does this benefit us, right? So one thing I noticed, as it was noted too, I originally in my PhD started in the cancer field, and one thing that we have in the cancer field is that we have great models. In fact, a lot of great in vitro models, especially cell culture models, because cancer, you basically you have a problem of trying to kill the cells. So when you put them in a dish, they just keep multiplying. They never stop. You don't have a problem culturing them. The heart is an exact opposite problem. The heart is a terminally differentiated post-mitotic cell type in the cardiomyocytes. So when you put them in culture, they only last for a day or two max, and then they all die. They don't divide. So you can't really keep them in culture. So that really makes it hard to do any mechanistic studies to really understand what's happening in the heart. So going back to this idea about the metabolic omnivore, what we did was we gave mice 13 seed traces or heavy labeled 13C non-radioactive isotopic tracers of different metabolites to figure out how the contribution of each fuel, different fuels, to the TCA cycle. And what we found is that about 23% of the TCA cycles coming, intermediates are coming from carbohydrates, 10% about or so coming from ketone bodies, but the vast majority are coming from fatty acids, which is not surprising. What is surprising is that when you break down the carbohydrates specifically, what you find is that only 17% of those TCA cycles coming from glucose. 83% is coming from lactate. So the vast majority of the TCA cycle and ATP that you're generating from carbohydrates is coming from lactate, not glucose. And yet, when you grow these cardiomyocytes in culture, you put them in media that basically has glucose, a buffer, and nothing else. It's more similar to something like Kool-Aid. It's not even close to being something physiologically relevant. So when you grow cells, these cardiomyocytes, primary cultured adult cardiomyocytes from mice, when you grow them in the regular DMEM without lactate, it basically just glucose and buffer. Within 3 days max, all your cells are dead. If you put them in something a little bit more physiologically relevant, something called HPLM, human plasma-like media, a media that was developed by Jason Cantor, which basically he took human plasma, ran it on a mass spec, and then saw what was in that plasma and made a media that reflected that. But you remove lactate specifically from that media, you can extend the life of these cardiomyocytes about to a week, but they still all die by the end of the week, even though they have fatty acids, even though they have ketone bodies, even though they have glucose, but they don't have lactate. Now, what was very interesting is that if you just add lactate, you can then improve the survivability of these, drastically improve the survivability of these adult cardiomyocytes. So if you have this DMEM, which is just glucose, and you give it 25 millimolar of glucose and you give it just 1.6 millimolar of lactate, you can extend their lives to over a week. You still have about 30% of your cells. But then if you have HPLM, with the fatty acids also, with lactate specifically, you can have your cells alive for over a week. And this is just a comparison of all the different media that you can buy commercially. And if you notice here, most of the media you buy really don't have a lot of things in them. They have like limited metabolites within them. No wonder your cells don't like them and can't really live very long with them, within them. And none of them have lactate except for HPLM. Moreover, when we did heavy label tracing in these cardiomyocytes, so here what I'm showing you is glucose heavy label, which means that you have 13C glucose rather than 12C, so it's a little bit heavier on a mass spec. You can tell the difference between the two. And glucose has 6 carbons, so all 6 of the carbons are heavy. And why is this cool and important? Because then you can follow and trace where the glucose is going and see how much of it is going to the TCA cycle, for example, and can look at M+2 because when this 3-carbon pyruvate gets into the TCA cycle, you lose 1 heavy carbon to CO2 and only 2 go into the TCA cycle. So you always look at M+2 because that's how much is coming from glucose. If you grow the adult cardiomyocytes in media without lactate, what the point I'm trying to make here is that you see almost 40% of the TCA cycle is coming from glucose. So a lot of the TCA cycle is being supported by glucose and in a context without lactate. However, if you give it lactate, that plummets down to less than 10%, which is more reflective of what you see in vivo. So in this context, the only reason that cardiomyocytes in culture are taking a lot of this glucose is because you give it no other option. The only fuel you're giving it is glucose, so it artificially rewires its metabolism to take this glucose to feed the TCA cycle because it has no other option. But if you give it lactate, it always preferentially utilizes lactate over glucose, which is more reflective of what you see in vivo. And this might explain why a lot of the in vitro experiments that you do in cells never reflect what you see in vivo, because you're putting them in this artificial environment that is not reflective of what has actually happened in physiology. So all of this is cool and important, of course, but it led us to a simple question of how is lactate utilized in cardiomyocytes? And surprisingly, not a lot of work has done because of what I just mentioned, or of this bad reputation that lactate has. Some people just assume it's bad, so they don't even study it as much. But nevertheless, what we did is that in mice, we gave them heavy labeled 13C lactate and followed where this heavy carbon lactate went into, how it fed into the TCA cycle. Now, the dogma in the field is that when lactate enters the cell, it gets converted into pyruvate. And then once it's converted to pyruvate, it enters into the mitochondria through the MPC, the mitochondrial pyruvate carrier. So we had already had the MPC knockout. So we said, okay, when we knock out MPC, how does that really affect the ability of lactate to feed the TCA cycle? And to our surprise, there was no difference. So here in red, we have the MPC knockout, and as you can see, this carbon was getting into the TCA cycle, feeding succinate and malate, and there was no difference between it and the wild type. So that suggesting that this lactate was getting into the mitochondria in a MPC independent manner. We then corroborated this with adult cardiomyocytes, and in this case, we know what's in the media because we make the media and we just give it heavy label 13C lactate, whereas in vivo, you don't know what's happening because it can always turn into something else. And again, we saw no difference between the knockout and the wild type. Now, this is very puzzling because lactate does not have the ability to cross the outer and inner membrane of the mitochondria on its own. It can't just diffuse through. It requires a transporter to actively push it in. So this means there has to be another transporter other than MPC that's facilitating this because there's no other way that lactate can get in. And this is not something that is known, okay? So nobody thinks of lactate as a metabolic fuel, but I thought, let me check the literature, see if anybody ever mentioned this. And it turns out back in the 80s and 90s, this great exercise physiologist, George Brooks from Berkeley, had suggested that MCT1, this is the monocarboxylate transporter 1, this is known to localize on the plasma membrane. He suggested that it could also localize in the mitochondria. Now, unfortunately for him, he was completely ignored and even ostracized, and nobody ever mentions his work in any textbook or review article that you read. You'll never find anything about MCT1 in the mitochondria unless it was written by George himself. And this is crazy because if you look at Mitocarda, so this is a database that was developed in the Broad Institute by Vamsi Mutha, where it basically saw every protein that localizes in the mitochondria. If you look in their database, they actually have MCT1 and LDHB within that database. And yet still nobody believed George or took his work seriously. Nevertheless, we thought we'd look into it more carefully, see if he was correct. So what I did was isolate mitochondria from our MPC knockouts and our MPC wild type, the flux. So you see here, looking at the levels of MPC. MPC here is low or gone. There's basically nothing left of it versus the wild type. And if you look at MCT1, what you see is that you see MCT1 in the mitochondria, but it also goes higher. It increases in the MPC knockout, which is very interesting. So this was the first suggestion to us that, oh, wait, maybe MCT1 does go to the mitochondria. Now, how do we show this? So we then thought, all right, let's image it. Let's see if it actually happens. So we did this microscopy with confocal laser scanning microscopy, looking at the mitochondria in red and MCT1 in green. And what we found was this very interesting pattern where you see the mitochondria here in red. And you see that when you do this line analysis, you see these MCT1 on the edges, but if within the mitochondria. And then if you look at the knockout, they're completely gone. Now this, if you don't know, suggests that the MCT1 is localizing to the inner mitochondrial membrane potentially. But this is not enough, evidence to show that. So let's look more carefully. So then we looked at something that we know for sure is in the inner mitochondrial membrane. and that's SLC25A6, and this is the ATP translocase 3. This is known to be in the inner mitochondrial membrane, and we see perfect co-localization between MCT1 in green and this protein that is known to be the inner mitochondrial membrane. And if we compare it to another protein that's known to be in the outer mitochondrial membrane, specifically here TOM20, which is known to be on the outer mitochondrial membrane, we see that they do not co-localize. So this suggests that it is in not only in the mitochondria, but in the inner mitochondrial membrane, which it has to be in order for this to mean anything. But that's not, is still not enough evidence to prove this. So we did the gold standard in the field. And the gold standard in the field is this proteinase K protection assay. Now, proteinase K is an enzyme that can digest any protein. However, it cannot penetrate the mitochondria. So it can't, anything outside of the mitochondria will be digested. So if it's something that's just sticking to the outside of the mitochondria, this will eat it up. So then we can differentiate between things that are sticking on the outside versus things that are inside. So for example, here we have ATP5A, which is known to be in the inner mitochondrial membrane. You see that this protein is completely protected throughout until we swell and digest and remove the membranes themselves. While something like TOM20, which is known to be in the outer mitochondrial membrane, every time we add proteinase K, the protein goes away because it's on the outer mitochondrial membrane. And then when you go look at something like citrate synthase, which is in the matrix, which is really in the core of the mitochondria, it's completely protected throughout this assay. And when you look at MCT1, you get the same exact pattern you get from ATP5A, definitively showing that MCT1 localizes to the inner mitochondrial membrane. So that's great. So then what happens if we knock it out? So then we repeated our experiments, our tracing experiments, and we developed these, instead of the MPC heart-specific inducible knockouts, we made MCT1 heart-specific inducible knockouts, and repeated with the 13C tracing lactate. And in this case, we see a significant decrease in the amount of carbon getting into the TCA cycle. Not only that, when we look at the adult cardiomyocytes, we also see this decrease only in the context of MCT1 knockout, not the MPC knockout, and inhibiting the MPC on top of the knockout didn't have a further decrease in this inhibition of getting the carbon inside the TCA cycle. Moreover, when we do when we harvest adult cardiomyocytes from these knockouts, they have no benefit from lactate. So lactate cannot extend their lives within culture if you don't have MCT1. So this benefit of lactate seems to be MCT1 dependent. And then to take it to another level, what we did is that we isolated mitochondria. So we got these mice, took their hearts, isolated mitochondria, and see if we can see any changes in the respiration and the oxygen consumption of these mitochondria from these different knockouts. So here, when we give them pyruvate, we see that there's a significant decrease in respiration in the MPC knockout, which is not surprising because MPC is what's letting pyruvate get into the mitochondria. However, there's no difference between the MCT1 and wild type, so pyruvate can get in no problem it doesn't require MCT1. However, if we switch the field from pyruvate to lactate, now we see a significant decrease in the MCT1 knockout, but not the MPC. So again, the MPC knockouts, they can readily respire lactate. And remember, these are isolated mitochondria. There are no other enzymes. Whatever is happening just from these mitochondria. But to take it to the next level also, what we did was 13C tracing. So not just oxygen consumption, but actually looking at the levels of the metabolites within the mitochondria. And if we give them 13C lactate, we also see a significant decrease in the amount of lactate feeding the TCA cycle only in the MCT1 knockout, but not the MPC. Now, this is all cool and interesting, except this was all in mice. And I know many of the audience will be wondering, what about human patients? So one of the cool things about Utah and working with Stavros is that we had a lot of human patient samples from both failing and non-failing hearts. And what I did is took some of these non-failing hearts, isolated mitochondria from human patients, and then treated them with an inhibitor that inhibits either MCT1 or UK5099, which inhibits MPC, and then see whether or not I see a difference from these isolated mitochondria from human hearts from 13C lactate. And as you can see here, we only see a significant decrease in the amount of carbon getting in when we inhibit MCT1, but not MPC. So this shows that this also happens in human patient hearts. But then the question is, why would the heart want to do this? What's the point? What's the benefit of any of this? Which is a good question. So one of the things I mentioned before was that the heart is a metabolic omnivore. And one of the observations that have been were made decades ago was that the heart can simultaneously take up glucose and lactate at the same time. It's known that it can do glycolysis, and it's known that it can uptake lactate at the same time. Now, the problem with this is that there are huge consequences for each one of these metabolic pathways. First of all, in glycolysis, at the last step, you go from pyruvate to lactate, and you generate an NAD. This NAD is necessary and important to keep glycolysis going. So the enzymes within glycolysis require this NAD to keep going. However, if you take lactate into the cell and then use it in the cytoplasm and go from lactate to pyruvate, you're going in the opposite direction. And instead of making NAD, you're consuming NAD and making NADH. And it's known that an increase in NADH levels will completely shut down and inhibit glycolysis. So how can the heart do both at the same time? It doesn't make sense. So what we think is happening is that if by directly importing the lactate into the mitochondria instead of utilizing it in the cytoplasm, you are now compartmentalizing and isolating this NADH within the mitochondria so that it does not affect the cytosolic NAD-NADH ratio. And so lactate can be consumed and utilized within the mitochondria while glycolysis continues in the cytoplasm at the same time. So then we want to show that this actually happens. So so instead of using 13C heavy carbon lactate, what we did was then we did this deuterated heavy hydrogen lactate. And the cool thing about this is that it's that this heavy hydrogen can then be taken by NADH. So when you do this reaction with LDH, lactate to pyruvate, this heavy hydrogen goes from NAD, is taken by NAD, gets NADH, and you see this heavy NADH that can only be coming from this heavy deuterated lactate. It doesn't happen normally. And when we did this experiment, what we saw is that in adult cardiomyocyte, there's a significant decrease in the amount of this heavy NADH in the MCT1 knockout. And if we looked at isolated mitochondria, we also saw a significant decrease in this heavy NADH in the MCT1 knockouts, but compared to the wild type. All right, that's cool. That means that we can get NADH from this reaction, but there's another consequence for this. So if we are correct and NADH lactate is being utilized within the mitochondria, we are suggesting that this is a new, novel way to import NADH into the mitochondria. So previously, what you look in textbooks, the main ways to get NADH into the mitochondria is by the malate aspartate shuttle or the glycerophosphate shuttle. There's no other way to get it in. So if lactate is indeed being utilized in the mitochondria, what we thought would, to prove this, is that if we inhibit the TCA cycle by using this inhibitor CPI-613 that inhibits PDH and alpha-ketoglutarate dehydrogenase, so inhibits the TCA cycle in two different parts, that if you give it lactate, the mitochondria should still be able to respire, to consume oxygen. And that's because if this is happening, if there is a generation of NADH, this can then be utilized by complex 1 of the electron transport chain to then pump that proton out and keep the ETC going. And these mitochondria should still be able to respire from lactate specifically, but not another fuel. So to test this, we gave them these isolated mitochondrial pyruvate, and when you give them CPI-613, there's a significant decrease in its ability to consume oxygen to respire, which is not surprising because we're inhibiting PDH and the TCA cycle in general. But if you give them lactate, there's no difference. So that is an incredible finding that lactate can can allow mitochondria to continuously respire even in the absence of the TCA cycle. And to even confirm this, we did 13C lactate with this inhibitor, and what you see is this carbon does not make it to the TCA cycle. So even though this carbon is not making it here, these mitochondria are still respiring just based on this mitochondrial NADH generation. So what's the physiological relevance of all of this? You know, this is all really cool observations. And this is an important question, because what we initially saw, and surprising to us, was that when we knocked out MCT1, even 1 year post the knockout, there was no significant phenotype, or at least no cardiac phenotype, that we could detect between the knockout and the wild type. Along everything, ejection fraction, the size, whatever we looked at, there was no difference. Now, if you remember in the MPC, when we knocked that one out, that was sufficient to induce hypertrophy, but in MCT1, there was no difference. So we thought, when I thought about it a little bit more deeply, I thought this probably was because of a redundancy between MPC and MCT1. So when you knock out MCT1, lactate, in theory at least, could still be converted to pyruvate and enter the mitochondria through MPC. And perhaps that was why these mice didn't really have a strong phenotype. And another thing that led me down this rabbit hole of thinking was that Zolt Arani at Penn had this great paper on Science, where he was looking at the contribution of different fuels in the heart, and with hearts of human patients with preserved ejection fraction and reduced ejection fraction. And what he saw was this unexpected finding that lactate contribution to the ATP actually doubled in the heart failing and failing hearts with the reduced ejection fraction. This was not expected, and he couldn't really explain how this is happening. So we looked at, again, patient samples, and what we found was, interestingly, that these MCT1 levels were actually increasing in heart failing patients. So these heart failing patients and donor non-failing patients, when we isolated their mitochondria, there was actually more MCT1 in the failing patients' mitochondria, which is very interesting. Moreover, if you remember from the first part where I was talking about how MPC can induce hypertrophy, what we found in that paper was that if you induce hypertrophy, that was sufficient to reduce MPC levels. So we thought, what would happen if we induced hypertrophy in the context of the MCT1 knockout? Could we generate a situation that's resembled a double knockout, where MCT1 was knocked out, we induce hypertrophy and MPC goes down? And what would happen if there was no other way for this carbohydrate to get into the mitochondria? So again, to do this, similar to the first paper, we surgically implanted these osmotic pumps with vehicle or angiotensin-epinephrine, kept them for 6 weeks, and then sacrificed with their wild type or MCT1 knockout. And to our surprise, what we found is that there was this accelerated heart failure in the MCT1 knockout. So you see this significant decrease in ejection fraction in the MCT1 knockout, specifically within 1 week. Now, if you do these experiments, what you know about the heart is that it's very resilient. Normally, you have to do this experiment for a month or two before you see any kind of phenotype. Here, within just 1 week in the MCT1 knockout, they immediately plummet, and you have this low ejection fraction suggestive of heart failure in these mice, and almost half of the mice die. We did 13C lactate tracing in adult cardiomyocytes with hypertrophy with the same exact treatment. And what we saw was that there's an increase in the amount of lactate contributing to the TCA cycle in the hypertrophied cardiomyocytes. And this fits what Zolt had seen with this increase in lactate contribution to the ATP. When you do this in adult cardiomyocytes, you see the same thing, like they're consuming more lactate because there's no MPC, so they have to do that. That's the main way to get in. But with the MCT1 knockout, there was no difference, which is not surprising because lactate can't get in. We also did the deuterated lactate experiment in this context as well, and we saw also an increase in this deuterated NADH. So there's an increase in this NADH, so that one of the consequences of getting more lactate in, you get more of this NADH, which is important. However, you don't get that in the knockout. And then we thought, okay, let's look at a different injury. So people had reported in the past that during ischemia or perfusion, there's an increase in MCT1 expression, but it's not known why that happens or what's the benefits of that. But so what we did was that we just, in our MCT1 knockouts, induced this ischemia or perfusion injury. Basically, we did an LAD ligation, 30 minutes, and then untie ligation, and then let them go for 120 minutes of reperfusion, and then ended the experiment. And what we saw there was that there was a significant increase in necrotic tissue and a significant decrease in salvageable tissue, even though the area of at risk was similar. So they had the same kind of ischemia tissue, but the difference, area of injury, but the difference was only in the necrotic tissue. There was more of that in the MCT1. So losing MCT1 was problematic in this model as well. And then when we looked at a slightly different model, what we did was that we did this LAD ligation, and then instead of doing a reperfusion for 2 hours, we just let them for 9 weeks. So let's see if the heart can actually recover. And surprisingly, what we found in the wild type, you have this initial dip in this ejection fraction, but over time, 9 weeks, they all recover, they get better, these mice. However, in the MCT1 knockout, you have this initial decrease, and it stays low, and it continues to go low, and you have this significant increase in their death, so that there's more than half of the mice die from this intervention. So the MCT1 seems to be very important in keeping this flexibility of the heart, and if you remove it, these mice cannot survive. So then, just quickly, just to wrap up a few last slides, there's still a lot more to discover. So even though this looks very intimidating and comprehensive, this doesn't even scratch the surface of what's actually happening in your cell. And then one thing, if you take nothing from what I've said, is that you have to always think about what's in your media. There's this paper that showed that, talk about how lactate is was not found in the mitochondrial fraction, and then lactate is not known to be a mitochondrial fuel. And this came from my paper. So I actually wrote this because what happened was that I was growing these cells in media that did not contain lactate. So I was not able to detect lactate in mitochondria because there was no lactate in the media to begin with. So you always have to think about what's in your media and not just what's in your wallet. Now, moving forward, so just quickly, just to talk about what my current lab and where we want to take all of this. So one of the cool things is that I'll be able to transport all of this, what I was talking about, into my own lab. And the big picture question is that how does lactate metabolism influence cellular decisions within the heart? And initially, at least, the three major projects I want to focus on is to continue a continuation of this mitochondrial MCT1 project in healthy injured hearts. Then look more at the cardioprotective role of lactate, and then lactate metabolism and lactoylation. So I'll just talk briefly about each in one slide for each one of these. As I mentioned, the MCT1 can get to the mitochondria, has huge consequences, but there's a lot more questions than answers. For example, how does MCT1 go to the mitochondria? We don't know. And how does it communicate with other ways of getting mitochondrial NADH, as I mentioned, like malate aspartate shuttle or the glycerol-phosphate shuttle? We don't know that, and so I will continue this work, and I will be able to bring all of the mice I mentioned from Utah to my lab here at Rutgers. So we'll still have these mice, and we'll still be able to ask these questions. The second one was very much influenced by this unexpected finding that lactate can extend the life of cardiomyocytes. These are notoriously difficult cells to work with. Nobody uses them. Nobody likes to work with them because they die within a day or two. They're a complete waste of money and time. But yet, if you add lactate, you can actually make them useful. So to me, at least, the logical step beyond this is not just to look at cardiomyocytes, but look at the heart itself. So the big picture idea here is that can we improve the solutions we use to transport hearts that are meant for transplant? Because, as many of you know, that a lot of these hearts that are meant for transplant make it to the OR and they're already damaged. just from that transport from one hospital to another. And the solution we currently use is basically just a buffer with glutathione and not raffinose and not much else. So what would happen if we added lactate and other important metabolites? And can we make it better so that every part that's meant for transplant can actually be used and be useful in the clinic? And of course, these are all things that I would require a lot of collaborations with clinicians like you guys. So that's why I'm pitching it to you. And the last one was to talk about this connection between metabolism and epigenetics. So in my PhD and in the cancer field, it's well known that metabolism has a strong, changes in metabolism can have a strong influence in epigenetics and change gene expression. So if you influence the metabolites that are used as substrates, you can drastically influence the histone modifications and gene expression of specific genes. And this is well known in the cancer field. In the heart field, nobody basically works on this. And moreover, there's this very specific histone modification known as histone lactoylation, where you have this lactate moiety on a histone that can increase his gene expression, very similar to acetylation, for example, but instead of an acetyl group, you have a histone group. Now, initially, at least, I would want to look in the context of hypertrophic cardiomyopathy. Now, this is a well-known genetic disease with variable penetrance and expressivity of the disease. So you can have two, for example, two siblings with the same exact mutation, but only one of them show the disease and the other does not show the disease. So obviously things beyond that mutation is controlling the disease itself. So could metabolism, histone modification have something to do with this? And we have some preliminary data to support this. So in the mice that I have, if we manipulate lactate, where you're looking at specifically the H3K18 lactoylation in vehicle, you have some lactoylation of general lactoylation happening. But if you inhibit lactate export, there's an increase. If you induce hypertrophy, you have an increase. And if you do both, you induce hypertrophy and inhibit lactate export, you have even more lactoylation happening. But what are the consequences of this? I still don't know. Moreover, when I grew these H9C2 cells in the presence or absence of lactate, one very interesting thing when we did the RNA-seq experiment in the volcano plot is that the genes that had the most change, that had the dramatic most change from the presence or absence of lactate were sarcomeric genes that are known to be mutated during hypertrophic cardiomyopathy. And this was very unexpected because I was thinking that including or removing lactate would probably influence metabolic genes, but that's not what happened. The ones that changed the most were sarcomeric genes. So what's the consequences of this and how does it relate to hypertrophic cardiomyopathy? Something I would love to work with any clinicians who have myectomy samples from hypertrophic cardiomyopathy or have their own ideas. And last but not least, at the end, I would just want to make sure that if you learned anything from this, is that lactate is not toxic, it's not trash, it's not a bad metabolite. If anything, it's got to be the best metabolite out there. My son tells me that God tier is not the best, that S tier is the best, but right now our funding can only support God tier, so maybe in the future we can go higher than this. And then acknowledgements, I have great collaborators at the University of Utah. Hopefully, my goal is to get as many great collaborators here at Rutgers. I've had great experience working with clinicians. I have a lot, if you look at my all of my publications, I always have a lot of multiple authors, and there's always clinicians within there. I think it's very important to work basic scientists with clinicians. And I got the opportunity actually to meet George Brooks, so the guy who actually first talked about MC2 being in the mitochondria, and he was so elated because he had actually been ostracized by the community at Hull, and he was very elated and happy that some random kid working out of Utah has proven correct after 30 years. And then I'm hiring. So if you know anybody that's interested in lactate, or if you yourself are interested, please apply or just email me and let me know. I'd love to work with you. Or if you just want to collaborate, that's even better. And with that, I'll take any questions.
Thank you very much, Ahmed. That was amazing. I think I have a series of questions for you. And I think I'll encourage participants to. I think this is an opportunity to do translational research I will encourage our fellows and our faculty who traditionally have done clinical research do cross-pollination and maybe design studies that can be done, maybe animal studies to humans or humans to animals, cell culture, whatever. So I think let's think out of the box here. So one thing I wanted to ask you, so we obviously have our heart transplant people and they're here and we can talk. We have a shock program. We can talk about how we could do there. But two or three areas which I immediately come to my impact is, I don't know if you know, we do sometimes very high risk PCIs, percutaneous coronary intervention, where we are using supports to unload the heart, because when you inflate the balloon, for example, if it's a main obstruction in the left main and all, the LV, if it is damaged, the ejection fraction will go down and the cardiac output will go down. I'm wondering, I don't know, this is a simple study that can be done. If your fluid or solution is so important, and if it should work, you could use we have catheters which are transport catheters, and maybe if there's somebody from interventional cardiology can work, where there's a balloon, but at the same time it can also continue to have injection through the lumen distally. So if you continuously feed that lactate rich solution, you could, I don't know, theoretically, you could probably reduce the impact of ischemia in those situations. Maybe we should do that in an animal model first and
Yes.
try to bring it to a clinical model. But we do a lot of high risk coronary work where ischemias are transiently created. And if there's a way for us to ensure metabolism can continue, or somehow train that myocardium by somehow to switch it to use metabolites differently while you're working on the coronary, maybe the patients will have better preservation. I also think you should think about cardioplegia solutions. For example, when you do coronary bypass surgery, you're putting the patients off pump, and basically there's a preservation of the myocardium. And one thing which I always see is that the right ventricle is very thin, very susceptible to ischemia. It gets stressed, it gets done, but it comes back very quickly. It's always alive, but it creates a havoc in a perioperative period. So how do we better conserve myocardium? These are all immediately coming to my mind, and these are all clinical questions that can be used to design impactful research, because it translates immediately to patient care. And of course, I would encourage people to latch on to any of this question, do anything that is needed. We also have Dr. Bakari creating a hypertrophic cardiomyopathy center, and there will be a lot of collaboration possible. But question to you is, have you you mentioned that you have successfully collaborated with physicians and fellows. We have tremendous large number of fellows. Chart out tell me a plan of how you will excite them and get them in. What will be the average? They look by the average churn time to a paper production. So tell me, how could they be churning and be salivating while they are taking a journey through you? I'm just throwing it out there. A lot to chew at.
Yeah, so there's a lot of opportunity. So one of the major opportunities I would say is that for me, from an outsider coming in, one of the major problems I saw is that a lot of the papers, when you read in the heart field, they're very observational. They just tell you we saw this in mice or we saw this in humans, and there's no mechanistic explanation of how or why that happened. Now, the beauty of a system like adult cardiomyocytes, for example, and now that we can actually extend their lives in culture, is that we can do a lot of mechanistic experiments and actually understand what is happening in these, what's causing the problem, and how, and when we are able to rescue, how are we rescuing it? What's actually happening on a molecular level? So we can now have this very strong, unique model that nobody else is doing right now. Nobody else is using our papers under revision now. So eventually people will know about this and, and use it themselves. But we have this great opportunity to start asking these very important questions and then having actually explaining how it's happening and why it's happening. There's a lot of interest. Like I feel the heart field now, especially in the metabolism field, is like where cancer metabolism was 10 years ago, where it's the hottest field out there. The cancer field now is cancer metabolism field is going down a little bit because it's getting so saturated. Nobody can compete in there. There's no more pathways left. But the heart field is open, wide open. We have this amazing metabolomics core. We have this opportunity to be the leaders in the field. Right now, there's like maybe 2 or 3 people in the heart field that do any metabolism that's really of high quality. Everybody just does something and not really understanding what they're doing most of the time, and they don't really have the core facilities to actually inquire about it. Because for example, I was on the job market very recently. I went to many places all over the country, and they always have like a proteomics core. Almost none of them have a metabolomics core. We have one that is one of the best in the nation, and I can tell this with somebody who has firsthand hands-on experience with doing metabolomics myself. This is one of the best out there. So we have an opportunity to become the leaders of this cell metabolism, cardio metabolism field, and connecting it to the histone modification is something that nobody's ever done in the heart field that I know of. So there's some people who already, like Maha Abdulatif, that's in the Newark campus in Rutgers, who's an expert at histone modification changes, but nobody's connecting the two yet. So we can be one of the first people, the pioneers, to do this before everybody else. Because once this, the first paper comes, then everybody is copycats. So we want to be the first people out there rather than the people that follow.
So can I ask you how much time does it take to culture these cardiomyocytes and do the work? And do you need an approval for doing it?
So yeah, no, it's cool. The cool thing about this is that everything happens after the mice is sacked. So after the mouse is dead, we don't really need any kind of approvals after that. And what happens is that it takes about 45 minutes and on average to get to harvest cardiomyocytes from a heart, and then another about 45 minutes to culture it and on a plate, and then it's up to you what you do after that. So it's about 2 hours work to really get.
So on an average, it takes about 3 months to get an IRB approval. I'm just giving people an
yes.
You probably in the meantime have generated a paper already.
Sure. Yes. Of course. Yes.
Okay, so I'm just giving people a journey of how fast you can move through, and you could also probably not require a mice animal. You could probably get adult cardiomyocytes already available somewhere and design your experiments, and you don't need any IRB approval, ICOOK approval for those kind of experiments. And you could design. I mean, it's a question of like the magic of how you think and what you can do. It's limitless, and the amount of productivity. I almost feel like I should just become a postdoc.
Sure.
The reason being, it's so exciting. Like, I mean, we didn't have these capabilities. I'm just exploring and I'm exciting people to think differently about research. So you do a small work, prove something exciting, and then try to go and try to show it in humans. In that way, you have imaging, you have Dr. Yanmala with her AI magic and large data sets. This is a good ecosystem for doing good, exciting research. I wanted to excite. One thing I'd really want to come to, and I want to make sure. So I want to talk to you, tell everybody about journey of an academician, right? You are an academician, right? So let me ask you, so you must have gone across the markets all across the US. And why did you choose academics? You could have just gone into a large company and become like a double 4-digit salary. I don't know what is possible. And why did you go this route? And of course, you have a family, you have children, and you survived.
Yes.
You're surviving. So academic journeys are possible, right?
Of course.
Explain to us because what I'm saying that tell us what keeps you. What is your lactate? How are you surviving despite not having any oxygen?
Yeah. Well, for me, the thing that's always been the reason that really attracted me to this way is this the intellectual freedom that you get, where you can just basically ask a question and just do whatever you want, and you're not beholden by a specific thing. When you're in a company, the end goal is to get a drug, and that drug has to be profitable. So you might find an actual drug target, but if it's not profitable, they'll kill the project right away. Very short-sighted, because they're looking for quick gains now. It turns out that if you continue studying that drug target, it turns out to be something very important that you were unexpected finding that explains something that was unknown and unconnected to what you were looking at initially. So this is to me, the beauty of it. And then you have this huge impact. Now you guys have hands-on impact on the patients. You get the luxury of actually seeing the impact of your work. But I would argue that you could have even a huger impact if you actually make the discovery that helps all the other researchers and doctors all over the world now know the answer, for example, of how to treat hypertrophic cardiomyopathy, because we did the experiments here and we figured it out.
Yeah, so I think, Dr. Yanmala, that's the reason we got Ahmed into your section. So I think we are very, to you now, I think I don't want to monopolize the conversation.
No, Dr. Sengupta, you have summarized it, the importance of bedside to bedside and bedside to community, right? I mean, this is the exact realm where we can connect from one spectrum to the other. And thank you for such an engaging discussion, Dr. Ahmed. One question I had was, like, when you talked about the localization of lactate in the cytoplasm versus mitochondria, all I was thinking about was cytoplasm is all about rapid anaerobic ATP production versus sustainable in the mitochondria for aerobic ATP source utilization. So one thing that kept bothering me was how does the other pathways like fatty acid oxidation and the shift, how does the shift during the different types of stress like hypoxia versus hypertrophy? And that was kind of, and then its relation to ischemic versus non-ischemic cardiovascular diseases was going in my mind. And did anybody study that?
No, see, that's the beauty of this. So a lot of what was done. So for those who don't know, like the whole field of metabolism was pretty much born in the heart field. So all the major findings in the initially, like post World War II and so on, it was all done in the heart field until the cancer field was born and people figured out how to culture cancer cells, and then everybody in the metabolism field moved to cancer, and the field of the heart field became a field that had almost nobody working on metabolism because it was very difficult. We didn't have the models. So a lot of your questions are really important and seem to be, you'll be surprised of how little we know about them. Because when you go and look in the publications, you'll find all of these great papers that came out in the 80s and 90s, and some of them in the 70s and 60s, but then not much recently. And then the more, and so how these different metabolic pathways communicate with each other is a lot more difficult to figure out when your only model is a mouse, because you can't tell what's happening, because you have on top of the heart, you have this circulation that happens between it and other organs, where it makes it almost impossible to figure out what's coming and what's going. And so you need something simpler and more easy to manipulate, like cell culture. And for those who in the metabolomics world, cell culture is like the best model they love, because they can control everything. You can control everything that is exposed, that the cells are exposed to, everything in the culture, the oxygen, everything you can control. Whereas in the mouse, you can't control anything. You don't know what's happening. And that's why there's always variation from mouse to mouse too. And you can't really control for that. Unlike in a cell culture model. And on the same note that you were talking about, so we recently, completely independent of anything I talked about today, figured out that, for example, LDH, the main enzyme that converts pyruvate to lactate or lactate to pyruvate, there's two major ones, A and B, and it seems that both can bind to ATP and both can bind to fatty acid, but only LDHA is inhibited by them, while LDHB is not. Now, why is that important? Because it turns out the major LDH in the heart is LDHB. It's actually called the heart type, because it's more or less the only LDH you find in the heart is the LDHB. And from our recent work, the only LDH in the mitochondria is also LDHB. So that makes sense that it cannot be inhibited by fatty acids and cannot be inhibited by ATP, because there's a lot of fatty acid oxidation happening. There's a lot of ATP happening. Now, during hypertrophy, more recent work by others has shown, and ourselves as well, is that LDHA goes up. Now, why is that happening? And what can we then now manipulate our knowledge and of metabolism to turn that into a drug target? Now, can we inhibit this LDHA that we know is inhibited by high levels of ATP and high levels of acyl-CoAs? So there's a lot that we don't know. So this, even though like that big metabolic pathway I put in the beginning that people are scared of, looks very comprehensive, it's really a very oversimplistic view of a metabolism. And what we don't know is we're barely scratching the surface. There's so much we don't know that's not even remotely included there.
So Ahmed, there's a lot of work at the moment going on in ischemic management of preconditioning. So I just was at TCT, and they're giving liquid oxygen, hyperbaric oxygen. I don't know what exactly is it in ischemia. So I think you should really look into that area, because there's a lot of companies around trying to work in that space. So if you have something, I'm hoping you're patenting some of this stuff, but designing some of these experiments, and if we can, I mean, this is exciting, because I think your body of work does show very convincingly that I mean, the lactate metabolism is fairly critical. At least it was very convincing in what you showed. So we are excited what will come out from you, and maybe you should design with Dr. Yanmala series of more exposures. Or maybe you might have to do like the space exposure that you have those missions. So we'll have our people transported in batches to your places, so that somebody will get excited and
Sure,
I have this responsibility to make sure my trainee environment and my young faculty who are doing a lot of clinical work do get the exposure and get the abilities to do cross-disciplinary work like this as well. It was not long back when Dr. Yanmala was exposed to the clinical community, and now she's a star, and very soon you will be a star as well.
On that note, Ahmed, so reminding everybody, what is the legacy you want to leave behind, right? In choosing academic path versus a professional track versus a industry job. On that note, I would like to thank you for giving us Grand Rounds today, and for such an engaging discussion as well, and for sharing your insights. Good day, everyone. Thank you.
Thank you very much.
Thank you. Appreciate that.